Hi everyone, I'm Sven Bultreis from Longevitas Consulting. At Longevitas, we help companies, investors, physicians, and scientists navigate the rapidly evolving longevity field by building the longevity ecosystem and translating cutting-edge science into actionable innovations and successful companies. Today, I want to tell you about an exciting new paper published in Nature Communications by researchers from Revel Pharmaceuticals. This study describes the development of an enzyme that can break down one specific type of advanced glycation end product, or AGE. If this approach can eventually be translated into the clinic, it will represent an entirely new strategy for targeting age-related protein damage. So what are AGEs? AGEs are formed when proteins and other biomolecules react with sugars or other reactive carbonyl compounds in a process called glycation. Our cells are quite good at preventing this damage by removing many of the reactive intermediates. However, once the final AGE products have formed, our bodies have essentially no way of reversing them. As a result, they gradually accumulate over the course of our lives. AGEs are especially abundant in people with diabetes, because elevated blood glucose accelerates their formation. They are thought to contribute to many diabetic complications, including cardiovascular disease, but AGEs also accumulate during normal aging and have long been proposed as one of the mechanisms that drive age-related tissue dysfunction. The authors focused on one particular AGE called carboxymethyl-lysine, or CML. As the name suggests, CML is formed when a lysine residue within a protein becomes chemically modified. The key insight behind this study came from looking closely at the chemistry. As you can see here, the modified portion of CML closely resembles the amino acid glycine. The researchers therefore wondered whether an enzyme that normally acts on glycine could be engineered to remove this age-related modification, ultimately creating what they called a CMLase. Their first experiments were encouraging. A bacterial enzyme called glycine oxidase from Bacillus subtilis successfully reversed free CML back into lysine. However, there was an important problem. Inside the body, CML is attached to proteins, not floating freely. And when the researchers tested CML attached to a peptide, the enzyme no longer worked. Several other glycine oxidases showed the same limitation. The researchers reasoned that the enzyme's active site simply wasn't large enough to accommodate a peptide substrate. To overcome this, they searched for related enzymes with different structures, using AlphaFold's predictions as a guide. This led them to a glycine oxidase from Calidothermus roseus, which showed a small amount of activity against CML-modified peptides. It wasn't good enough, but it was a starting point. The team then turned to directed evolution, a powerful protein engineering technique that mimics natural evolution in the laboratory. They began with an E. coli strain that could no longer produce its own lysine. These bacteria could only grow if lysine became available from the environment. Next, they generated thousands of mutated versions of the enzyme using error-prone PCR and expressed these variants in the bacteria. Any mutant enzyme that more efficiently modified CML back into lysine gave the bacteria a growth advantage, allowing the researchers to rapidly identify improved variants. After the first round of evolution, they obtained an enzyme containing 7 amino acid substitutions that showed roughly a 5-fold improvement in activity. Following 4 additional rounds of directed evolution to improve activity, substrate accessibility, and stability, the final enzyme contained 15 amino acid substitutions and 2 amino acid deletions, with more than a 10-fold increase in catalytic efficiency compared with the original enzyme. Next came the really exciting part. The team showed that CMLase efficiently removed CML from several biologically relevant proteins, including albumin, casein, collagen, hemoglobin, and proteins extracted from the eye, reducing CML levels by between 52 and 97% after overnight treatment. They then moved on to human tissues, where AGEs accumulate over decades. Treatment of arterial tissue from elderly individuals reduced CML levels by about 70%. In elderly skin, CML levels were reduced by approximately 55%, bringing them down to the levels similar to those found in the tissue from a 31-year-old individual. These are striking proof-of-concept results showing that an engineered enzyme can remove an age-associated protein modification directly from human tissues. That said, there are still several important hurdles before this approach could become a therapy. First, we don't yet know how effectively this enzyme will penetrate dense tissues in a living organism. Second, because the enzyme is derived from bacteria, it could potentially trigger an immune response. Additional engineering may therefore be needed to reduce its immunogenicity. Third, this work has not yet been tested in living animals, so its safety, delivery, and therapeutic benefits remain unknown. Finally, it's important to remember that CML is only one member of a much larger family of AGEs. One particularly important AGE is glucosamine, which is actually the most abundant protein crosslink found in aging human tissues. During my internship in the laboratory of Professor David Spiegel at Yale University, I worked on the chemical synthesis of glucosamine. Having synthetic glucosamine available could eventually help researchers develop similar enzyme engineering strategies. However, unlike CML, there currently isn't an obvious natural enzyme that recognizes a structural similar molecule, making directed evolution considerably more challenging. Finding a suitable starting point remains an open question. Overall, I think this is a fascinating study. It demonstrates for the first time that directed evolution can produce an enzyme capable of reversing a specific age-related protein modification in biologically relevant proteins, and even in human tissues. Although significant work remains before this could become a therapeutic intervention, it opens an entirely new avenue for developing enzymes that repair molecular damage associated with aging. If you enjoyed this breakdown of recent scientific publications, let me know. I'd love to make more videos like this in the future. Thanks for watching, and I see you next time. Bye bye.